Hi, welcome to my super high-tech biology lesson for today. This week we are talking about mutations and how they can affect a cell. And it completely builds off of what we did last week in protein synthesis. And so if you understood what we did last week in protein synthesis, this week should be a piece of cake. But if you did not understand protein synthesis, this is not going to make a whole lot of sense. So I might hit on some of that protein synthesis again. So first and foremost, what in the world is a mutation? Well, a mutation is any change in our DNA. And I know that there are a lot of images that might come to your mind for mutations like the Ninja Turtles or X-Men or I don't know what other pop culture mutation thing might be out there, but Spider-Man. But there would have to be a lot of mutations that take place in order for an organism to change so much in that particular way. But a mutation is any change in our DNA. And there's a really high chance that you have some mutations. Whether or not they show a physical trait is a whole nother thing. So a mutation is any change in your DNA, and there are ways that those mutations can affect your traits if these happen in genes. And so just a reminder that a gene is a segment of DNA that codes for protein. And I'm going to introduce a new vocab word. We have allele. I think there's an E on the end. An allele is a various type of gene. So if you have a gene, but there are different kinds of that gene, that would be an allele. So different variations of the same gene. And there is a likelihood that variation arose because of a mutation at some point. So if a gene undergoes a mutation, then you are going to see a variation of that gene. All right, so why does this matter? Well, recall that we have body cells. And in all of our body cells, inside the nucleus, we have DNA. And remember that inside, we have 46 chromosomes. That comes from 23 pairs, meaning we got 23 from our mother and 23 from our father. And on each chromosome, you have these places that have genes. So those bands that I just drew represent a gene, okay? And you got, if this is chromosome 1, just for an example, you got a chromosome 1 from your mother and a chromosome 1 from your father. And they have the same genes on them, but they might be different variations of that gene. So if you go back and think of middle school when you did those Punnett squares, remember Punnett squares where you did big B, little b, crossed with little b, and you might have learned about Gregor Mendel, and you're really excited because it's fun puzzles, but doesn't always make sense. This is what we're talking about here. These letters we give to the different variations of the genes that are available. So on these chromosomes that you got from mom and dad, they might have the same variation. They might have the same allele for that gene, or they might have different alleles. How about this one here? Let's make this one big B and little b. Now those have different alleles, okay? So these alleles are formed oftentimes by mutations. So we're going to talk about how mutations occur and the different types of mutations that you can have. I'm changing color pen. Okay, so there are two main types of mutations. You have point mutations, which are also known as substitutions. Or you have frameshift mutations. Okay, which are caused by insertion or deletion. You like my high-tech notes? They're super fancy. Okay, so substitutions. Let's start with a genetic code, okay? I have one printed out here because I have a codon chart that goes with it. So let's look at these mutations, okay? We're looking at substitutions first. All right, so I have this genetic code, and last week we learned that the first thing we do is to transcribe the code, which means turn it into an RNA, and then we can translate the code to figure out what protein we have. Okay, and remember that you need this codon chart for translation. Okay, that's our tool that we're going to use for translation. All right, so decoding the DNA, we're transcribing it first. So T goes to A goes to U, C goes to G. I'm going to finish this strand. Remember, we use U's in RNA, not T's, so that's where that came from. All right, so remember that these are in 3-letter segments. That is our frame, that's our 3-letter frame, and we use this to translate. So AUG in the codon chart here is found right down here. So that is the MET, or methionine amino acid. It doesn't matter what exactly it's called. Just write down the abbreviation. That's fine for this activity. GAA is located right here. So that should be GLU. CCC is right here. That should be pro. And UUU right here. That should be PHE. Okay, I have 4 codons there, so I should have 4 amino acids, and I do. Okay, so that's my amino acid. So if we consider this to be the original, so if this is the original, what would happen if we were to mutate that DNA strand? And there are a couple different things that can occur. Okay, so the first thing let's look at is what happens if we were to change this C to an A. So let's try that. I'm going to use a different color since we're working on a different problem. If we change this C to an A, everything else remains the same, right? So transcribing will be the same for all of these, except now here, A will go with U. So let me change this to U. Okay, this is our mutation. We changed the DNA. So this is translated the same, but what is UAA translated into now? Well, if I go over to my codon chart, UAA equals a stop. So instead of glue, we have stop. Stop means to stop, game over. You cannot go any further. And so none of the rest of this will be translated, okay? The protein is literally cut off right there. And so a lot of times this is going to cause a dysfunctional protein. So we call this type of substitution nonsense. Nonsense mutations, come back. nonsense mutations give you a stop where there should not be one. Okay. So let's try a different one. Okay. If we keep the same code that we have up there, and I'm just going to move down to this one. Let's cross this off. Don't need that. All right. So if we keep all things the same here, except maybe we mutate this one. And again, it's just one base pair mutation. And we substitute it for a C. Okay? This is just one base pair mutation. This is just one base pair mutation. Just a substitution here. What happens now? Well, this is the same and this is the same. This one's going to be different. So let's translate. TAC is into AUG. CTT is GAA. GGC should be CCG. And AAA is still UUU. Okay? So this is the same. This is the same. But what is CCG? Remember, that's different than this one. So looking at my codon chart, CCG is pro. But wait a second. CCC was also pro. And then this is phi, just like the other one. So I have one amino acid, one base pair changed here, but my amino acid did not change at all. And this is called a silent mutation. So a silent mutation gives you no change in amino acid. Okay. And so let's look at one more type here. If we have a, let's see, So let's change this to a T. Okay, so instead of UUU, we now have UUA. So I'm just going to use this whole strand the same and change this to an A. And this is going to change this amino acid to a LEU. So in this example, every amino acid is the same, except this one has been changed into a LEU, which I think is leucine. So that we call a missense mutation. One amino acid change. So those are three things that can occur when you have a point mutation or a substitution. Now, there's another type of mutation called a frameshift mutation. And for a frameshift mutation, there has to be some sort of insertion or deletion, and it's exactly like what it sounds like. So if I write a code A, T, I'm going to try to make this easy to understand. So I'm going to make the triplets the same, C, 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 and G, G, G. Okay, so if this is our DNA code, and we transcribe it to RNA, we now have U, 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 and A, and G, 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 and C, C, C. Okay, and then we can go back to our codon chart and we can even translate for the different proteins. But what I want to show you is what happens when we have a shift. If we were to bring in a new amino acid or new nucleotide here and insert itself maybe right there, everything shifts over a little bit. Okay, so so this will remain the same, but then this shifts over to